Genetic crosses. What we're going to do here is just work through question 2 of the homework pack, and just to show you how to go about answering exam questions and a couple of different ways to approach genetic cross diagrams. So question 2 in the homework booklet states that there is a species of snail where the shell colour is controlled by a gene that has 3 versions or 3 alleles. So they're telling us that the 3 different phenotypes are brown shells, pink shells, and yellow shells. Now we're told that the allele for brown shells, or CB, is dominant to both other alleles. That is the most dominant allele. Then the allele for pink, or CP, is dominant to the allele for yellow, or CY. So the first thing to do is just get your head around the level of dominance that we have here. So you can do this on an exam paper, you can write it out. If you're looking at that, they're telling us that B is the most dominant. It's dominant over P and it's dominant over Y. They've also told us that P is dominant over Y. So you can see how they relate to each other. Okay? So the first question says, give all of the different genotypes, so the combinations of alleles, which would result in a snail having a brown shell. So again, come back to this. We know that the brown allele is the dominant one. So as long as they have at least one brown allele, they will have a brown shell. So what combinations might we have? Well, we start off with our one brown allele. That could be joined by a second brown allele. So you would be homozygous for the brown allele. What else could we have? We could have another single brown allele, and this time we could pair it with a pink allele. It's dominant over the pink, so you would still have a brown phenotype. The third thing we could do is have our single brown allele and combine it with a yellow allele. Again, the brown would be more dominant than the yellow, so it would still have a brown shell. So those would be the 3 possible genotypes, and they would all result in a brown-shelled snail. Hopefully that's quite straightforward. So now we move on to using a genetic cross to demonstrate how 2 pink-shelled snails, so 2 pink parents, could produce only pink-shelled and yellow-shelled snails. So their offspring can only be pink or yellow. So where do we start with this? First thing to think about in this example is, what are the parents going to be like in terms of their genotype? We know their phenotype is pink. Okay? We also know that they have yellow offspring. So we're looking at the pink and the yellow alleles here. We can't be looking at the browns because the brown would have dominated everything. So the parents have either got to be homozygous pink, so CP, or heterozygous for yellow and pink. Now, how do we narrow that down further? Well, we know that the offspring that are yellow have to have 2 yellow alleles. So that means one of the yellow alleles must have come from one parent and the other must have come from the other parent. That means both parents have to have a yellow allele. So quite simply, we know that they both have a yellow, definitely, but they both must also have a pink allele because they are actually pink in terms of their phenotype. So they've got to have one yellow and they've got to have one pink. So we instantly know that both parents are heterozygous, pink and yellow. And once we know that, we can move on to actually doing a genetic cross. Now, there are 2 different ways to carry out or draw out a genetic cross, and I'm going to run you through both of them. Okay, so the first one is probably, I think, the simplest. It's a Punnett square. So in a Punnett square, we start off with a blank table or a blank grid, doesn't really matter what you want to call it. And then we place our parents into that. So we've got pink parents, 2 of them, obviously, and we are making sure that we are including both the phenotype, that they are pink, and also the genotype, so which alleles they have. So we know that they are heterozygous, they have 1 pink allele and 1 yellow allele. Okay. Next, we start filling in the gametes. Okay, so those are the sex cells. So very simply, we just transfer the alleles down. So if you look here, we are taking that pink allele and placing it in here. So 1 gamete will hold that. And then the yellow allele we're placing into a different gamete. And we just repeat that for the other parent. Okay, now we look at those 4 blank white boxes. That's where we are going to place the genotypes and then the phenotypes for the offspring. Okay, now they've already told us that the offspring are pink and yellow, so we know that already. But how can we prove that? So again, we just transfer across those alleles into the boxes. So this first offspring, it's got a pink allele from one gamete and a pink from the other gamete, from the other parent. So that is homozygous. Okay, then we move into the next box, a pink allele from one parent, a yellow from the other, so that is heterozygous. Moving down to the bottom ones, we've got a pink from one parent and a yellow from the other, heterozygous. And then finally, we have our yellow allele from one parent and a yellow allele from the other parent, and we've now got a homozygous yellow individual. Now, the last thing we need to do is put the phenotypes into those boxes. So if you're in any doubt, look back to the grid that I gave you at the beginning, or the hierarchy that I gave you, so that you remember which is dominant over which. So we know that the pink is always dominant over the yellow. So if you work your way through, if you've got two pinks, you will have a pink shell. That is your phenotype. If you are a pink and a yellow, because the pink is still dominant, you will still have a pink shell. Again, another pink and a yellow will result in a pink shell. And finally, if you've got two yellows, you are going to have a yellow shell phenotype. So again, we've got the genotype in each box and we've got the phenotype in each box. If you were to do that, you would have got full marks for this question. So that is the Punnett square. There is another example that I'm now going to run you through. So this one is one that I was certainly taught at school, but sometimes it can look a little bit messy. So again, you start off with your two parents and you make sure that you have the genotypes and the phenotypes in those boxes. Then we work out the gametes. And again, it's just a case of taking one allele out of the pairing and putting it into a separate gamete. So we've got the pink, then the yellow, then the pink, and then the yellow. So we've got our 4 different types of gametes. And then it's a case of matching up the gametes. Now, obviously, you have to have one gamete from each parent and work through the different combinations. So for our first offspring, we would have a pink from the first parent and a pink from the second parent, homozygous pink. Okay, another pink from the first parent, this time a yellow from the second parent, so it'd be heterozygous. Then we're on to a yellow from the first parent and a pink from the second parent. We're on to heterozygous again. And then finally, we've got the yellow from the first parent and the yellow from the second parent. And we've got our homozygous yellow. And again, make sure that you remember the order in terms of the dominance levels, and then you can work out and place in the phenotypes. So homozygous pink is a pink shell. Heterozygous will still be a pink shell. Heterozygous, pink shell. And finally, homozygous for the yellow will be a yellow shell. You've got your genotype and your phenotype. Hopefully that made sense. Remember to go back through this if you need to watch it again and slow it down.